In pharmacology, understanding the mechanism of action of a new modality is critical to gaining the trust of patients, physicians and regulators. Investors and stakeholders want to understand the specific biochemical interaction through which a drug, candidate or combination therapy produces its pharmacological effects and also potential side effects. However, elucidating the mechanism of action of a drug, candidate can be quite tedious, as signaling networks are complex and interact with numerous other signaling pathways. Using validated cell lines, organoids or animal models, it is useful to analyze between 200 and 800 proteins for a comprehensive understanding of pharmacological effects, with particular attention to the phosphorylation status of regulatory proteins. Over the past decades, researchers have used ligand-binding assay platforms such as ELISA, electrochemiluminescence or Western blots, each with specific advantages and limitations. While most of these platforms have sufficient assay sensitivity and dynamic range, they are limited in terms of their multiplexing capabilities and ability to quantify phosphorylated proteins, as there is no sandwich pair for most phosphoproteins. While the direct assay approach of Western blots is perfect for quantifying phosphoproteins, analyzing more than 20 proteins is quite labor-intensive even when using ProteinSimple's platform. Hyperplex is the first ligand-binding assay platform that enables the analysis of more than 800 proteins and 300 phosphoproteins in a single run. Hyperplex uses commercially available antibodies from leading suppliers, such as cell signaling technologies. Let's look at how Bayer Pharmaceuticals uses Hyperplex to elucidate mechanism of action. Regorafenib and sorafenib are two approved cancer drugs from Bayer Pharmaceuticals. Since these small molecule drugs differ only by one fluorine atom in the central phenyl ring, it was assumed that both drugs had the same mechanism of action. Surprisingly, however, it was observed that regorafenib showed better activity in sorafenib refractory patients. The company investigated the matter in preclinical models of hepatocellular carcinoma using Hyperplex Protein Profiling. Hyperplex is an innovative, patented platform that combines the protein size resolution of gel electrophoresis with the unbeatable specificity of a ligand-binding assay. In a first step, a known amount of protein is size separated by automated gel electrophoresis, yielding exactly 96 protein size fractions. The proteins are biotinylated and immobilized on the surface of Nutravid-coated Luminex beads. A specific bead set is added to each of the 96 wells, resulting in a collection of distinguishable protein-loaded bead sets from a single sample. The beads are pooled to reconstruct the original sample, with a defined color code correlating to the respective molecular weight of the immobilized proteins. A small aliquot of the bead pool is incubated with up to 800 Western blot antibodies before the sample is detected on a Luminex FlexMap instrument. Results are tabulated and displayed as a heat map or volcano plot. Hyperplex offers a dynamic range of 3.5 orders of magnitude and can be combined with any antibody validated for use in a Western blot. The combination of automated electrophoresis with ligand-binding detection has several advantages. First, the direct ligand-binding assay format requires only one antibody, and since there is no sandwich pair for most phosphoproteins, Hyperplex is the only ligand-binding assay format that makes more than 300 phosphorylated biomarkers available in a single multiplex assay. Second, the combination of two very different protein profiling technologies adds confidence to the results. Finally, more than 1,250 antibodies from leading vendors, such as cell signaling technologies, have been qualified on the Hyperplex platform. This expert curated content is structured into more than 60 functional pathway networks and disease-specific panels. Users can easily customize panels of up to 800 protein biomarkers online, and newly developed proprietary antibodies can be added to all pre-qualified panels. Hyperplex analysis was performed on total protein lysates from drug and vehicle-treated PDX xenografts. Protein expression was significantly different in regorafenib and sorafenib-treated samples compared to controls. The pattern of upregulated proteins was similar with both drugs, indicating an activated RAF-MEK-ERK pathway, but more proteins were downregulated with sorafenib than with regorafenib. Overall, both regorafenib and sorafenib were effective in the mouse models, although in some cases regorafenib showed better activity, which may explain the observed efficacy of regorafenib in sorafenib refractory patients. Hyperplex proved to be a powerful tool for quantifying hundreds of proteins, providing a very comprehensive overview of the drug's mechanism of action. Multiplex phosphoprotein analysis provided actionable data on pathway modulation, including heat maps, volcano plots and pathway maps. Hyperplex project reports disclose all antibodies and their commercial sources, allowing seamless integration of Hyperplex data into sponsors' workflows. Hyperplex gives you instant access to more than 1,250 expert curated biomarkers, more than 60 pathways and signaling networks, and more than 20 disease-centric marker panels. If you are considering using Hyperplex for any of your projects, make an appointment with a Hyperplex scientist now.